Hi, so just a brief introduction to mu-opioid receptor signaling and the research pathways to reduce tolerance and withdrawal and maximize hedonia. So opioid receptors, all opioid receptors are what's known as G-protein coupled receptors, which just basically means that when they bind to their agonist and are activated, they activate something called a G-protein heteromer. And it's known as a heteromer because it's made up of these different subunits. You can see here the alpha, beta, and gamma. And there are many different gene types of alpha, and then within that, different splice variants of each of those genes. So there's so many different combinations of each of these three subunits to form many different G-protein heteromers with different properties. But that's the basic idea. You may also have heard G-protein coupled receptors referred to as 7-transmembrane receptors, or 7TM. And you can kind of see quite clearly in this diagram the reason for that is that the polypeptide chain crosses the membrane bilayer 7 times. So there are 3 main types of opioid receptors. There's the kappa, delta, and mu, and there are also some others like the opioid receptor-like or ORL. But for the purposes of this presentation, we're going to be focusing on the mu receptor because this is the most studied and it's thought to be the most responsible for analgesia, although the other receptors are somewhat involved. But that's the reason for the focus on the mu opioid receptor. So I've used the word agonist a few times already, and it just basically means any molecule which binds to a receptor and activates it, as opposed to an antagonist which would inhibit it or block it. So there are kind of 2 main types of agonists. You can have exogenous agonists, or like externally administered agonists like morphine and fentanyl. You might have heard of those as pain-relieving medication, or people take them recreationally. And endogenous, which are like enkephalins, endomorphins, substances that bind to and activate mu-opioid receptors, which are naturally produced in the body. And the obvious difference between these are that obviously your endogenous opioid peptides don't produce any side effects, you don't get tolerance and withdrawal from them, whereas with drugs like morphine you do. So a lot of research is focused on trying to work out the differences and why that is that morphine produces these side effects and the endogenous drugs don't, and whether we can try and find ways of mimicking the action of endogenous opioids to maximize hedonia and minimize tolerance and withdrawal. So we can do that either by producing altered agonists and synthesizing new types of ligands to bind to the mu-opioid receptor, or other downstream interventions, which I will discuss later. So just briefly, one of the signaling pathways which is activated by the mu-opioid receptor is G-protein signaling. And just to mention that this G-protein heteromer is only shown like this for clarity, but in reality it's actually bound in the membrane bilayer, as you saw in the previous diagram, by some fatty acid tails coming off the alpha and gamma. But for clarity we've just shown it here. So when the agonist binds to the mu-opioid receptor, it induces a conformational change which is propagated across the membrane bilayer and ultimately leads to activation of the G-protein heteromer. And you can tell that this is activated because it's bound to GTP. So when it's activated, GTP exchanges and replaces GDP, which would be in the inactive form. And this active G-protein heteromer complex will inhibit the adenylyl cyclase pathway. And adenylyl cyclase is an enzyme which catalyzes the formation of cyclic AMP, which is a second messenger which leads to activation of various kinases and various downstream pathways, which we'll discuss a bit more later. And another effect is this effect on potassium and calcium transport. So as you can see, the potassium channel is open by the active alpha subunit, and this leads to potassium flowing out of the cell into the extracellular space, down its concentration gradient, so from the area of high concentration to low concentration. And this flow of potassium ions can lead to hyperpolarization, which will terminate the propagation of an action potential. So it could ultimately lead to inhibition of a pain signal across a synapse. And the same goes for the closing of the calcium channel, which prevents calcium from flowing in. And calcium is another second messenger like CAMP, which stimulates the release of neurotransmitters by exocytosis at the presynaptic node. So inhibition of calcium import and opening of potassium channel ultimately leads to inhibition of a pain transmission. So that's what gives you your analgesia.